Hello guys, so today we are going to talk about deviation from Mendelism. What happens in Mendelism is basically we talked about Mendel's law of independent assortment, and there was Mendel's law of segregation. But there are a few topics that deviate from the laws of Mendel's, and they are not in accordance with the laws of Mendel. So what are these? These are incomplete dominance, codominance, pleiotropy, polygenes phenomenon, that basically interaction of genes, and then we have linkage. Today we are going to talk about epistasis, that is quite similar to polygenes phenomenon. Yeah, and incomplete dominance, we saw that if we had a white color flower and a red color flower, we had a mixture of them both as a pink color flower, and none of the white and red color flowers showed, and basically showed their colors properly, and there was no proper dominance from red or white, but there was a mixture, there was an incomplete phenomenon in the flower, and we saw the pink color. Next, we saw about the codominance. In the cases of blood groups, we had A and B blood group, and like if father has A blood group and mother has B blood group, we can have AB blood group in children. Yeah, and there were a phenomenon of polygenes. My last video at that. So let's talk about today's topic that we are going to talk about today is yeah, so this topic is epistasis. So Mendel thought that one factor controls one character, right? One factor or one gene control one character. At that time, Mendel didn't know about knew about genes, so he called them factors, and he said that one factor actually controls one character, one character. So one he thought that one gene is responsible for controlling the character or one trait. But in these days, we have found these phenomenons of polygenes phenomenon and pleiotropy, in which the phenomenons are very different, and we see that one character can be controlled by many genes in interaction of genes or polygenes, and can be one factor can be controlled by many genes, and many genes can work on one character or one trait, right? So these are the two opposite phenomenons. Then let's talk about yeah, so let's talk about dominance. So what is the difference between dominance and epistasis? So basically, when we talk about dominance, we say that, for example, if we had this gene, and here we have an allele A, and then on the next uh, chromatid, we have this allele small a. So here we said that capital A is the dominant one and small a is the recessive one. So this allele would be dominant, and this allele would be recessive. So it is basically interallelic process, because it is between two genes, and because it is in between only one gene, the process occurs in one gene, same gene alleles. So we say that this is basically an intragenic process. Okay, but when we talk about epistasis, then what happens in epistasis basically? So we have these capital A and small a, and let's assume that they are like big brother. Capital A is like big brother, and small a is like younger one, younger brother. Okay, so now what happens is that big brother always dominate the younger brother, and he sometimes beats him. He is like that bold character of the house, and uh, a younger one follows him, and he is the dominating one. Okay, so what happens is that both of them lived in the same home or same gene, but On an other gene, there are two other brothers that live there, and they are um, capital C and small c. So sometimes what happens that these sometimes if there are two younger brothers of other house, they will come and they will beat the bigger brother of this house. They can beat the bigger brother of this house and like the elder brother, sorry, of this house, and they will in dominate him, and their characteristic will be, or their trait will be represented or shown. Like let's see the next slide and we will get a more clear idea. Let us move on to the next one. So when we talk about epistasis, it's now, this effect is will be basically intergenic, because it would be between the genes, between two different genes, between two different homes. Now, the boys of two different homes are involved, and okay. So next thing is suppressing gene, the gene that is suppressing the other one. For example, these two guys of another gene of another home came and dominated over the elder brother of elder brother of our home, right? So basically, what we would say that yeah, we would say that they shouldn't have done that, right? So what would happen basically is that these two boys would be called epistatic. Okay, so basically these two genes would be called epistatic, and the genes that are getting suppressed would be called hypostatic genes. So this one would be hypostatic and this one would be epistatic. So if what happens that CC can suppress A, it's called recessive epistasis. It means that two younger siblings of another gene or of another home comes and dominate over this elder brother of gene 1 or home 1, right? But if the W can suppress Y. This is dominant epistasis. But if the uh, dominant allele of the other gene comes over and dominate the first gene of another allele of another gene, and both are dominant, then only one dominant of other gene can suppress the dominant of the this gene. So this happens, this is called dominant epistasis, and this one is called recessive epistasis basically. So now let's move further and let's talk about some, okay, now how polygenetic inheritance and epistasis are similar. Both are quite similar as multiple genes are involved in expression of one trait, but one factor that differentiates the epistasis from polygenic inheritance is variation. So discontinuous variation happens in epistasis and continuous variation happens in polygenic inheritance, because as in the case of height, we saw that if you have the height of 6 feet, there would be 5'9", 5'8", 5'7", 5'6". Okay, so now the variation is very continuous. But in the case of epistasis, we would maybe in the skin color of dog, we would see like 3 4 colors or at max. So these variations are not very continuous, these are discontinuous variations. Okay, yeah, let's move on further and yeah. So now we would talk about Labrador Retriever. So Labrador Retriever comes in these cute colors, black, yellow, and brown. So example, let's see. The interplay between these genes determine the color of Labrador Retriever, and what happens is basically is that black Labrador can have any genotype with at least one dominant allele at both B and E loci. So what happens is that, for example, in Labrador Retriever, there is this gene here, this gene with B dominant allele and B recessive allele. And there is another gene with E dominant allele and E recessive allele. Now, what happens basically is that if combination forms as such that there is one dominant in both of these genes, one dominant expresses itself in both the dominant genes, then black color would be expressed, right? But if what happens is that this VB phenomenon is expressed in any case, then if small b comes twice, then we'll be having the chocolate color or brown color Labradors. And the yellow Labradors can be found if there is two small e. So if there are two small e, no matter if there is two capital E on the other hand, or if there is capital B, small b on the other gene. So if these two younger siblings are coming together to beat the elder one, they will dominate. So yeah, this is the epistasis, and so this is the case of us epistasis and recessive epistasis to be exact, because here two recessive alleles are coming and they are dominating over dominant genes, dominant alleles. Okay, so let's move on further. Yeah, so here you can see that if we will have a cross. For example, we had this BBEE, and we made its cross with BBEE. Now we will be having BB and EE. So now in two generations, BBEE, we will cross together them together, and what will happen basically that we will be having like such combinations in which we have like nine basically black color dogs, and then um, we would have three chocolate color dogs, and the yellow dogs would be four in number, and that is basically epistasis. So epistasis happened in four of the dogs if we made this Punnett square of 16 people. So yeah, this is the ratio in epistasis. I hope you got the point. Thank you so much for watching, and take care. Bye bye.